Alrighty, let's get started with chapter 10.6. Alrighty, now today we're going to be talking about what controls the cell cycle. Now before we begin, we have some important overview questions, just review before we dive in. So the first one is, why is controlling the cell cycle important, right? Then the second one is, what are checkpoints and how many are there? What is the main engine of cell division? What are the functions of cyclosomes, and what are the ways multicellular organisms differ from unicellular organisms in terms of controlling the cell cycle. It is very important that the cell cycle is controlled, otherwise we can have diseases like cancer. The cell cycle is controlled during DNA replication, the separation of sister chromatids, and particular points in the cell cycle called checkpoints. During checkpoints, the cell cycle may be stopped if there is something wrong with DNA, and gives the cell time to respond to external and internal signals. The research over the control of the cell cycle is informative because it puts modern observations into context, and it shows biologists using very different methods but achieving the same results. The next 3 slides are of different observations, and later we will see how they all work together. Now, the discovery of the maturation promoting factor, which is MPF, was made by studying frog oocytes. Oocytes would become arrested at the G2 stage before meiosis I, where they needed hormones to signal the beginning of their division. The cytoplasm from cells that were currently dividing were extracted and put into these cells, which caused them to be prematurely induced into cell division. This pointed to there being a regulator of cell cycle progression, which we know as MPF. This experiment is depicted in the image. The discovery of cyclins was made by studying sea urchin embryos early in their division. Specifically, they discovered proteins that were made along with the cell cycle, with 2 different forms of cyclin discovered reaching peaks at the G1S stage and the G2M stage. There is no enzymatic activity in cyclins alone. The discovery of the CDC2 gene was made by studying yeast. After examining certain yeast cells that stopped division because of a mutation, the scientists discovered there are 2 points that have to be crossed in order to divide: the commitment to DNA synthesis and the commitment to mitosis. The gene CDC2, found in fission yeast, was found to be the critical gene needed for both thresholds. There are 2 key aspects of the maturation promoting factor, or MPF. The first is that its activity varies during the cell cycle. It starts off as low, slowly increases throughout the G2 phase, and climaxes during mitosis. Secondly, the MPF is involved in enzymatic phosphorylation of proteins, which is important because phosphorylation is reversible and allows the proteins to be activated and deactivated. The CDC2 gene provides the instructions to make a kinase protein, which combined with the cyclin will make an MPF. Upon that discovery, the CDC2 gene protein was named a cyclin-dependent kinase, or otherwise known as a CDK, and MPF was renamed to mitosis promoting factor instead. CDKs are the main controllers of the cell cycle and are often described as the engine of cell division. The cell cycle is controlled at 3 checkpoints. They're called the G1/S, the G2/M, and the late metaphase, or otherwise known as the spindle checkpoint. The G1S checkpoint is mainly when the cell chooses to divide or not, and is the main checkpoint when external signals influence the cycle. It is also the phase at which growth factors affect the cell, and the phase that connects cell division to cell growth. The G2/M checkpoint ensures the success of DNA replication and can pause the cell cycle if it is not done correctly. If a cell passes through this checkpoint, it is a commitment to mitosis. If there is an agent that is discovered damaging the DNA, the cell will be arrested. CDKs that are present here are shown as MPFs, which are now called M-phase promoting factors. The spindle checkpoint makes sure that chromosomes are attached to the spindle at the metaphase plate for anaphase. This step is very crucial because the separation of chromosomes is irreversible. So that means that it has to be very accurate and there needs to be proper alignment. As said before, the CDKs are the engine that drives the cell cycle, and they power the cell cycle through phosphorylation. Phosphorylation is going to be the main mechanism of cell cycle control. Not only that, but CDK activity itself is controlled by phosphorylation, and also the activity is determined by the number of cyclins. Full activation of the CDC2 kinase requires complexing with the cyclin and phosphorylation. MPF at the G2/M checkpoint is controlled by two things. MPF needs the presence of M-phase specific cyclins and the removal of an inhibiting phosphate group by phosphatase. At the checkpoint, damage to DNA may cause MPF to be inactivated as well. The purpose of the anaphase promoting complex, otherwise known as the cyclosome, is to trigger anaphase. For it does this by marking the protein securin for destruction. Once it is destroyed, the enzyme separase is no longer inhibited and can now destroy a part of cohesin. Once a part of cohesin is destroyed, that allows the sister chromatids to go to opposite poles because they are no longer together. I will show this in an actual hand drawing. Alrighty, so we start out with our APC, which is our cyclosome. So this is going to attach ubiquitin to securin right here. Ubiquitin, right? And so this proteasome is going to come by and see the securin with the ubiquitin and get rid of it and destroy it. It is gone. So now we have our separase, and our separase is no longer allosterically inhibited by the securin because it is gone. So now this is able to come here to the sister chromatids, and it is able to get rid of a very specific part of the cohesin, which allows the sister chromatids to spread apart like that. The cyclosome has two roles in mitosis. First, it activates the separase that removes the cohesins holding the sister chromatids together as previously mentioned. Second, it is required for the destruction of mitotic cyclins to get the cell out of mitosis. It does this by adding ubiquitin to marked cyclins for degradation by a proteasome. There are two major differences between the multicellular eukaryotes and single-celled eukaryotes concerning the control of the cell cycle. In multicellular organisms, there are multiple CDKs as opposed to the single CDK in single-celled eukaryotes. Also, multicellular organisms have a much greater amount of external signals to respond to. But despite that, the basic mechanisms of both are the same. For example, the human and budding yeast control of the cell cycle are both very similar in the way they work. In multicellular organisms, organization is maintained by only having certain cells divide at certain times. When mammalian cells grow in a tissue culture, a single layer of cells expand over the culture until it touches another group of cells. Once it touches another group of cells, it stops expanding because receptor proteins trigger a signal transduction pathway that inhibits CDK action. Growth factors trigger intracellular signaling systems for the onset of cell division. One of these growth factors is the platelet-derived growth factor, PDGF, which is a receptor tyrosine kinase that starts cell division. There are over 300 peptides with over 30 families of related peptides that have been identified to have growth factor activity, and a specific cell surface receptor recognizes each growth factor, which are often MAP kinase cascades that stimulate the proliferation of G1 cyclins and other proteins necessary for the cell cycle. G0 is the dormant phase of the cell cycle, whenever the cell has not passed any checkpoints. For cells that regularly divide, whenever a cell does not pass the G1/S checkpoint, it is arrested and disallowed from passing through the checkpoint, entering into the G0 phase. Different cells divide at different rates. Stomach cells divide more than twice a day, liver cells once every year or two, and muscle cells and neurons almost never leave the G0 phase. Alrighty, now back to our overview questions. Why is controlling the cell cycle important? Controlling the cell cycle is important because it helps to prevent a lot of diseases like cancers from happening. Another reason why it's important is because it allows for increased accuracy in replicating DNA and prevents bad DNA from being replicated. What are checkpoints and how many are there? So checkpoints are basically points in the cell cycle when the cell is stopped and it is made sure that all the DNA is accurate and all the DNA that is being replicated is good to go and that the cell is good to go. And there are 3 checkpoints, the G1/S, the G2/M, and the spindle checkpoint. What is the main engine of the cell division? The main engine of cell division are going to be the CDKs. CDKs phosphorylate everything, and that phosphorylation is basically what helps to control the cell. It's what mainly controls the cell. What are the functions of cyclosomes? Cyclosomes have 2 functions. The first one is to trigger anaphase by attaching ubiquitin onto a securin, and then once that securin is marked for destruction, and once it is destroyed, separase is allowed to separate the sister chromatids away from each other. The other function of cyclosomes is to mark mitotic cyclins for destruction. And finally, what are the ways multicellular organisms differ from unicellular organisms in terms of controlling the cell cycle? They differ in a couple ways. For one, multicellular organisms have multiple CDKs as opposed to the single CDK in single-cell eukaryotes. And also, multicellular organisms have a much greater amount of external signals to respond to. And here is where all of my pictures came from, all the websites that are listed, or the textbook where I got it from. So that's it. Have fun with the questions.